Let's get into the nitty-gritty now. Let's have a look at how we can use the fossil record as evidence of evolution. So there's a number of different types of evidence that scientists, particularly paleontologists, that we're going to focus on use in order to support the theory of evolution. Evolution is still considered to be a theory, so the more evidence that we can have to support the emergence of different life forms, the better that is for us. So we've got here a list of different forms of evidence. So we can use biochemistry, biogeography, which we don't look in this year's study design at anymore, comparative anatomy and embryology. So that's when we're comparing the structures of developing organisms to see similarities between different groups. The fossil record, which is a huge focus for us today. Genetic linkage groups and vestigial organs, which also fall under that heading of structural morphology. Okay, this graphic here just breaks down the headings that we just looked at. So we're going to focus today on the fossil record and how we can use absolute and relative age to date fossils to find out how old certain things are. We're also going to look a little bit at comparative anatomy, comparative embryology, and molecular biology as ways that we can date, with a focus on DNA sequence for that section. Okay, let's have a look at the fossil record. So fossils are evidence of past life. So I'm sure that you've all at some stage throughout your life spoken about and learnt about fossils. What we need to know is that fossils can be bones, they can be teeth, they can be parts of an organism, they can even be footprints. So we don't necessarily have to have a whole complete skeleton of an organism to have a fossil. We can have traces of those. So basically what happens during fossilization is an organism dies. So you can see a little purple dinosaur over here. That organism is buried by sediments. Its skin and everything is broken down by microbes, and the remains then turn into a fossil. Over time, the fossils flatten due to the compression or the weight of the layers of rock above it. The rock is folded and eroded, and the fossil can then be seen on the surface. So if over time in the history of our Earth, the layers of the Earth often move. For, when there's an earthquake, there might be a volcanic eruption, there might be some sort of seismic activity which leads to movement of parts of the Earth. But there also might be things like floods which wash away layers on the surface of the Earth and expose fossils. Okay, we can just see a little diagram popping up on the side here with some explanation arrows. But basically what we can see here is some rock strata, so some layers of rock. Within that rock, there's different fossils. The younger fossils are at the top, and the older fossils are at the bottom of those layers. Okay, which makes sense because the closer you are to the top of the surface, the more recently those remains have been fossilized compared to things that are right down the bottom. As we touched on earlier, there's a few different types of fossils. We can have physical fossils, which are physical remains of a skeleton, like bones, teeth, or leaves of a plant. Trace fossils. So trace fossils are preserved evidence such as footprints or tooth marks or tracks. So we don't necessarily have an actual skeletal structure, but we've got evidence of that organism being present on Earth a long time ago based on their footprint or tooth marks or tracks that they left or burrows. Organisms can also leave biosignatures or biomarkers. So these provide evidence of a past life and that may be inferred to exist. So these can be physical or chemical signs preserved in minerals. Okay, and for this one here, I often think of Jurassic Park when you've got the tree sap that's preserved which has preserved the mosquito, which had bitten a dinosaur and taken in some of its blood and therefore taken in some of its genetic material. So those are what we mean by biosignatures or biomarkers. We don't really need to focus on these guys too much for our exam, but it's good to have an idea in case we're asked a question about that. Okay, so in terms of those types of fossils that we just discussed, fossils can sometimes be macroscopic, so they can be seen with the human eye, or they can be microscopic, which means that we need a microscope in order to view them. Today we've also got some really great technology that enhances both macroscopic and microscopic fossils, using high resolution X-ray to create 3D images of these fossils. So we might be left with just parts of a skeleton, and with this technology we're able to recreate a 3D image of what that would have looked like as a whole. We've already spoken a little bit about how fossils are formed with those 4 different steps, with the organism dying, being covered by sediment, being flattened due to the pressure of those layers of rock over time, and then being exposed due to erosion. But there's some conditions that enhance the preservation of tissues and increase the chance of fossilization. So if we've got conditions which allow for rapid burial in sediments straight after the organism has died, so such as silt deposits on a lake or sediments on the seafloor, ash from a volcanic eruption. So the quicker something is covered, the more likely it is to be able to fossilize successfully. Burial in conditions that are alkaline or oxygen depleted, like into basically a pool of mud at the bottom of a stagnant pool. If an organism remains falling to those types of areas, they're going to fossilize really well. And also if there is an occurrence of death in a really cold environment, fossils can also be preserved by freezing. So think of in the middle of Antarctica, it's very, very cold. If an explorer, for example, was to accidentally slip, hurt himself, fall down and pass out, he would die from hypothermia because there is no one out there to save him, and his remains would basically be frozen. And because they're frozen in layers of ice, they're able to be preserved that way. Okay, this is really important and often. Okay, this is just a little graphic here which shows chemical traces and preserved remains of different structures over time. So have a little bit of a look at that and a read through the dot points at the bottom, because it helps us just to get into context a little bit, the age of different organisms that we're going to potentially have to look at. Okay, let's unpack those 2 ways that we date fossils a little bit more. So the first one we'll look at is a fossil's relative age. So there are 2 methods that can be used to identify relative age of rock. The stratigraphic method, which is based on the principle of superposition. So if we have a look at this particular picture up here, we can see the oldest rock strata, or the oldest layer of rock, is at the bottom, the youngest layer is at the top. So if we're able to label those layers and look at what fossils are found in those layers, compare them with another piece of rock from another area, we're able to work out a relative age that way. We can also use index fossils, and index fossils are fossils that were short-lived, so they only appear in one layer of a rock strata. So we can use those to compare to other fossils that we've found to try and work out a relative age that way. When we're looking at absolute age, what we're looking at is the rate of breakdown of a couple of known elements. So it's really important that we have an understanding of this table. So what we're looking at basically is finding out absolute age by looking at the amount of a radioactive parent isotope. So I'll take the first row of our table here, where we're looking at carbon-14. Carbon-14 will actually change into nitrogen-14 over a known rate of time. So you can see, and you'll see on the following slides as well as the Ed Rollo videos, that we talk about half-life. So a half-life is the amount of time it takes for half of the carbon-14 that's present in our sample of rock to change into nitrogen-14. Okay, that half-life, or the amount of time that it takes for that to happen, is 5,730 years. Okay, so we know that if half of our carbon has decayed, that we've got a range of 5,730 years that we're looking at. Okay, when we're using this method where we're looking at carbon-14 and it's changed to nitrogen-14, we can really only date fossils up to about 60,000 years old. So we use this for dating relatively recent fossils. There are other types of absolute dating or radiometric dating that we can use where we look at other elements. These other ones are when we're looking at fossils that are quite old compared to those ones that we look at when we're using our carbon. So we use uranium and look at its breakdown into lead over time. The half-life of uranium-235, so the amount of time that it takes for half of this to break down into lead-207, is 710 million years. So this particular technique would be used to date fossils that are 10 million years and older. Okay, as we move down our list of different radioactive parent isotopes, the amount of time or the date range that we can use is really, really long. So in terms of what we would see on an exam, we would probably see the use of these two techniques all being where we would need to mention those two as being the most effective in looking at the age of a fossil. Sometimes on an exam you'll get a question that says a fossil is, 29 million years old. What type of radiometric dating or what type of isotope would we use? In that particular case, we would look at something like potassium-40, okay, because the uranium-235, that's 10 million years and older. The potassium-40 is used for dating igneous rocks containing particular materials that are 0.5 million years and older. So how does the fossil record support evolution? So if evolution has occurred, we can look at the change in a fossil over time to look at how a particular species has evolved. Okay, so if we look at the change of this modern day horse over time, we can see that it's started off like right down the bottom here with in its foot, its lower foot. Basically, it had 4 toes. Over time, due to its change in its environment, we can see that it's now got a hoof. But we can look at those fossils over time to help us to help provide us with evidence that that's how this organism, that's what its journey has been over time to get to where it currently is. Also, teeth are another really good example. So these modern day horses started off with very small molars, and over time the size of the molar has increased to the large molar that's present today. And that will be due to environmental pressures and what food sources that particular organism has got. Also, as the organism has gotten larger in size over time, it's going to need more food to be able to power its metabolism. So having larger teeth makes sense because they're going to be grinding through more grass. We can also see transitional fossils. So transitional fossils are fossils that basically exist in one organism that show the emergence of another organism over time. So we can see here how we've got basically a skeleton of a dinosaur or a dinosaur type organism, and how that has changed over time into a modern day flying bird. So you can see that obviously its tail has been lost to an extent. It's still got a short tail, but not as much as it originally did. You can see that it's still got the cap at the back of its head. You can see that its body has become much smaller. Its rib cage has changed in size to help to be able to allow it to be able to fly and be a bit more streamlined in the air. But it's still got the wishbone to help to form its wing. So transitional fossils provide evidence or relationships between one ancestral species and another. There are some problems with using the fossil record. As you can imagine, it can be scanty. So the number of fossils compared to the number of previously living organisms is minuscule. So there's not many fossils to give us enough evidence or a record of change over time. As we saw before, conditions need to be right to have fossilization occur, and that's not always possible. Even in a correct environment for fossilization, bones can be disturbed by waterways, by scavengers, just by decay over time. It can't be said that 2 fossils come from the same member of the same species because they can't interbreed, so that's another complication. And generally fossils are not complete, and it can be difficult to put pieces back together as shown in the pictures below. Okay, so this one here, although we've got technology that help us to do this a bit better, it's still not 100% accurate to what the organism was initially.